Hello everyone, this is Soumya and today I'm presenting the prevalence of inappropriate image duplication in biomedical research publication. So this is the agenda for today. So the study determines the percentage of published papers having inappropriate image duplication on a specific type of inaccurate data. The authors have examined over 20,000 papers from 40 different journals having a period from 1995 to 2014. These papers contained the term Western blot, which is a scientific technique used in cell and molecular biology to detect specific proteins. These papers were all visually screened. Out of these 20,000 plus papers, 3.8% had inaccurate data. Now, your inaccurate data can occur either due to honest error or intentional falsifications. Your honest error constitutes incorrect calculation or using a wrong reagent, but intentional falsification is due to deliberate misconduct. So, why is the study important? This inaccurate data results in societal injury. There was a study associating measles vaccine with autism. This created a lot of resistance among people, which may have contributed to low vaccine rates. Other part is the contribution to research reproducibility. This research reproducibility in pharmaceutical industry caused termination of drug developments too. Oncology, out of 53 landmark papers, only 6 could be reproduced. So how were these images identified for duplicacy? These images were divided into 3 categories. So the first category is simple duplications. Here, 2 or more identical panels were copy pasted within the same figure or in different figures of the same paper. But these two figures represented different experimental conditions. So as you can see, the green panels here, they're exactly the same, but they are from different experiments. This could be due to honest error or misconduct. In the second category, a blot of image is either overlapped by rotation or shifting. So if you closely look at the green portions in this image and follow the pattern, it is exactly the same. This is mostly due to a misconduct. In the third category, the image is altered with complete or partial duplications of lanes or bands within the same image. So it can be rotated and duplicated. So this is also completely duplicated. So these two bands. Okay, so the results and findings. So we know that approximately 1 of every 25 published papers contains inappropriately duplicated images. As stated by the author, this is an underestimated value and could contain data anomalies as well. Since only image data were analyzed, so the errors in numerical data, tables, or graph is still unknown. There could be more, but we are still not aware. Only duplicated images within the paper were examined. So if there was an image in one paper and there was a duplicate in the second paper, those were not checked for. This issue rose sharply after 2002 and has since remained at increased levels. So though the cause of this is unknown, but the author believes that availability and usage of digital image modification softwares may have caused this. Even increased competition and career-related pressure may have encouraged carelessness. Hence, you see a sharp rise after the 2002. The fourth point states that it varies among journals and correlates inversely with the journal impact factor. That means that impact factor, the higher impact factor of the journal is inversely related. The journals having a high impact had low percentage of papers having these issues. So higher impact journals are better able to detect anomalous images prior to publication. It may be that authors submitted more carefully because they have a high impact, or maybe the journals had a proper policy in place. Okay, so the next is it originated more frequently from China and India. In this particular study, only papers from PLOS 1 were taken into account. It is suggested that scientific ethics reform should pay more attention here. Other papers by authors of papers containing inappropriate duplicated images often contain duplicated images as well. So, at least in 40% instances in which a problematic paper was found, the author repeated that particular type of mistake or duplicacy in another papers as well. The methods used for conducting the study was purely manual. They would visually check for duplications independently and classify only if all three authors agreed. They sometimes used Preview, which is a Mac software, for brightness adjustments to detect better. In statistical analysis, they leveraged the R tool, and as you can see here, this is the linear regression model. It shows negative correlation between the impact factor and papers with problematic images. The gray area is 95% confidence. So the pros for the study was that it definitely created awareness and was in the top 25 of 2016. It was also controversial because it resulted in at least 62 corrections and 6 retractions, which is a good in scientific industry. It also highlighted the need of scrutiny over publications. It is important to note that the scrutiny over publications is required across all publications. If one of them is lenient, the researchers may tend to lean towards that publication and get their papers published quickly. So the cons, with pros, we have cons as well. One major cons that I felt was visual limitations and no use of software tools. I assume if they leveraged a software tool, maybe they could analyze double the number of papers they have analyzed right now. And it took them 2 years to do this analysis. Only duplicated image within the same paper were examined, not a duplicated image across two different papers. If a paper that had multiple issues in multiple figures, it was not weighted more than a paper with only one issue. It was weighted at a similar level. So the evaluations. The first evaluations that I found was DARPA's Media Forensics, which is the MEDIFO program. It assesses the integrity of an image or a video. It checks if the image or a video is duplicated and even lets you know how it got duplicated. An interesting fact is that this tool used the data and images from this particular paper for its testing. The second tool that could be leveraged was forensic tools. So they are based on Adobe and Photoshop and can be used for detecting manipulated images. It is quite famous in the biomedical world. The thirdly, I found one paper which is automatic detection of image manipulations. So it depicts an entire software pipeline of just right from scanning the paper as a PDF, converting it into JPEG, and clipping out the image portions and detecting the manipulated portions in it. This paper also used data from our paper for its testing. Fourthly, I found USC's Deep Fake Detector. It is funded by DARPA's MEDIFOR program. And what it has done is it has leveraged artificial intelligence to detect manipulated or fake videos, which have 96% of accuracy. Next, towards our related coursework. So our coursework did have deduplication and similarity and why detect near duplicate slides. While we have not used any software methods to detect similarity of images in this particular paper, but this paper does show the impact of detecting near duplicates. It did find corrections in papers and forced people to get them corrected. So it is a good initiative and definitely it had an impact. Next, I have a fun video for you guys. So I hope you enjoy it and detect some similarities. Interestingly, this video was shared by this paper's author, Elizabeth Beck, so I could not miss putting it in this presentation. These are the references. Thank you.